Good morning, children. Now, for 10th standard, we have already taken the lesson, that is lesson number 18, genetics. In this, we have completed 2 pages. Who is Mendel? Then, how he came to know the father of genetics, and what are the things, how he can prove his research by using the pea plant. The other name of pea plant is Pisum sativum, I said botanical name, and he has crossed the characters, different characters of the pea plant. And that is monohybrid and dihybrid, everything I said. And which is the important allelome of alleles, everything we have seen. Then which is the important thing, that is Mendel laws, based on the experiments, based on his theory, what are the laws proposed by him. That's otherwise known as laws of heredity, laws of dominant, law of segregation, law of independent assortment. Now, which is the important factor for the main, that is genetics gene, where the gene present in the chromosome. That too I said. Now we have to see the structure of chromosome. It's in page number 265. Okay. The diagram what you are saying is structure of chromosome. So this is outer cover. The outer cover is known as pellicle. This is outer cover of the chromosome. This is known as what? Pellicle. This is pellicle. Clear, children? Listen, clear? So you have to label the parts very clearly. This is pellicle. So here the chromosome constricted in 2 sides, that is become narrow. That is, the first one is primary constriction, second one is secondary constriction. Actually the chromosome is 2 arms. This is one arm, this is one arm. Both the arms are joined together. Okay, inside the chromosome you can see this kind of the double layer. I made only single, double. Okay, so this is known as spindle fibers. This is known as what? Spindle fibers. Double layers should be there. In between the layers, you can see this is double layer, I said no, this is double layer. In between the layers, you can see the dots. The dots are known as what? Chromonimata. The dots are known as chromonimata that can be seen all over the chromosome. Okay, so what are the things? And last position, last part of chromosome is telomere. And some of the chromosome, you can see that ball-like structure, knob-like structure. This is known as what? Satellite. So this kind of chromosomes are known as satellite chromosomes. So we have to see the structure. You have to draw the diagram. So in the biological sense, explain the chromosome with structure. Structure means what? You have to give the diagram. So what is it? This is telomere. The starting point is what? The ending point is telomere. Outer cover is pellicle. And there are 2 constriction, primary constriction and secondary constriction. Inside the thing, this is one arm, this is one arm. Both arms are connected together. And this is double layer is known as spindle fiber. Within the spindle fiber, you can see the small dots. That is known as chromonimata. Now let us see in page number 265, the structure of chromosome. Page number what I said, 265. Clear? Now, this is important 5-mark question, children. So understand very clearly. The chromosomes are thin, long and thread-like structures. They are thin, long and thread-like. It consisting of 2 identical strands called sister chromatids. This is one, 2 arms. They are held together by the centromere. They both are connected by what I said. This is centromere. The both are connected, primary constriction, the connected layer, they are connected by centromere. Primary constriction, other one layer is there, that is centromere. Okay. Point by point you can write. How you can write? The chromosomes are thin, long and thread-like structure consisting of 2 identical strands called sister chromatids. Second, they are held together by the centromere. Okay. Each chromatid is made up of spirally coiled thin structure called chromonimata. This one, dots. The chromonimata has number of bead-like structure. Bead means what? The money in like structures along with its length, which are called chromomeres. The chromosomes are made up of DNA and RNA. DNA and RNA, they are the nucleic acid which brings the hereditary characters. DNA I said already, deoxyribo. Deoxyribonucleic acid. This is RNA, ribonucleic acid. Clear? The chromosomes are made up of DNA and RNA, chromosomal proteins and certain metallic ions. These proteins provide structural support to the chromosome. So all our internal parts, certainly that should be the protein which helps for the growth of the organism. A chromosome consists of the following region. So you have seen the outer structure of chromosome. What are the parts inside that? Primary constriction. That is, two arms of the chromosome meet at a point. They are known as primary constriction. This is one arm. This is second arm. Both are meeting at a point. That is known as what? Primary constriction. This line is primary constriction. And it is covered by a layer that is centromere. Primary constriction or centromere. The centromere is the region where spindle fibers attach the chromosome during the cell division. This and all, they are dividing together and joining together during the cell division, which helps the plant to develop the cells of the organisms. Secondary constriction. Some chromosome possess secondary constriction. The secondary constriction you can see only certain chromosome, not in all the chromosomes. It maintains and provides the stability to the chromosome. It gives the support to the, they are known as nuclear zone or nuclear organizer. The secondary chromosome is otherwise known as what? Secondary constriction is otherwise known as nuclear organizer. Telomere, that is end of the chromosome, is filled with telomere. Each extremity of the chromosome has a polarity and prevents it from joining the adjacent chromosome. This telomere just disconnect the joining of the chromosome. Chromosome may join together. One chromosome, another chromosome join agi. So to avoid that, what is there? Telomere. The chromosomes are the important factors. We have some changes in our body, because we are normal. But some you can say six fingers will be there. Sometimes we have seen in the papers 2 heads together. That is only one body, 2 heads. This is because of chromosomal distribution. While they are dividing abnormally, means we have different characters in our body. Clear? So satellite, this is only for this. Some characters, some cases of chromosome, this is satellite. Some of the chromosome have an elongated knob-like appendages at one end of the chromosome known as satellite. The chromosome with satellite are called sat chromosome, special kind of chromosome. Structure of chromosome is very, very important. Just go through that. Now, we say centromere. Based on the centromere, we can divide the chromosomes into type of chromosome based on the position of centromere. Position of centromere means what? Where it is, which place the centromere is. So, based on the position of centromere, chromosome can be divided into position of chromosome. Based on the position means what I said, where it is. Telocentric, acrocentric, submetacentric and metacentric. So based on the position of centromere. Position where the centromere is placed, position on that telocentric. Then acrocentric. And next one is submetacentric. Metacentric. Based on the position of centromere, chromosomes can be divided into 4 kinds, that is telocentric, acrocentric, submetacentric and metacentric, where the centromere is placed. We have seen already chromosomes are 2 hands joined together, the 2 chromatids joined together. Here see telocentric, centromere in one corner, here like this, and what is there? Chromosome. This is what? Telocentric. That is centromere in the corner, one corner. There is no chance for the second hand. Then acrocentric, it is somewhat developed. This is one hand is short. This is centromere. Other hand is long. This is one arm. This is one arm. Both the arms are connected by thing. One is short. Other one is long. This is acrocentric. And submetacentric, it is in the shape of L. That is, centromere is found near the center of the chromosome, thus forming two unequal arms. Here also somewhat developed. Arm is somewhat developed like this. This is one arm, and this is centromere, and this is other arm. This is what I said. This one is big. Two unequal. And the metacentric, both are equal. Centromere in center, here one arm, and here clear. Types of chromosome based on the centromere, it can be divided into 4 kinds of chromosome: telocentric, acrocentric, submetacentric, and metacentric. Telocentric, the centromere is found on the proximal end. They are rod-shaped chromosome. Acrocentric, centromere is found at one of the end. Other 2 arms are there. One arm is small, other one is long. Submetacentric, the centromere is found near the center of the chromosome. Thus, they forming 2 unequal arms. They are in L-shaped. Clear? Submetacentric, the centromere occurs in the center. The chromosome are formed into 2 equal arms. They are V-shaped. Clear? The structures are given there. Then types of chromosome based on the function. This is position of the centromere. So first we have seen chromosome. Chromosome further divided into many kinds. That is, one is position of centromere, other one is based on the function. Okay? Based on function, it is known as autosome and allosome. Autosome, we have 2 kinds of chromosomes in our body. That is, totally 23 pair of chromosomes in human beings. Then 22 pair autosome. Only 1 pair is allosome. Auto means somatic cells, that is bodily cells. Autosome, bodily cells. Allosome, reproductive. Further, we can divide the chromosome. Due to function, we can divide them into autosome and allosome. That is, autosome, they are helpful for the development of the body, the cell division for the thing, and only 1 pair of chromosome that will help for the reproduction, that is allosome. Clear? So, in the male we can see both kind of chromosome. That is, in the male we can see X and Y. In the female we can see X and X. So, this chromosome, this allosomes, the chromosomes only determines the sex of the new organism. Either it is a male or female, that can be determined by the chromosome. Suppose X and X, that is X chromosome of male and X chromosome of female, means the sex will be what? Female. X and X means female. Then Y from male, X from female means male. We used to say that is by God. Certainly, this is naturally occurring, but God helps through the determination of the chromosomes only. This position, function of the chromosome, they were divided into autosome and allosome. We have 23 pair of chromosome, 22 autosome, body chromosome, only one allosome, that is reproductive chromosome. Male has 2 kinds of chromosome, X and Y, female X and X. This chromosome determines the sex. That is, sex means what? What kind of baby it is. What kind of, even animals also. Karyotype. Karyotypes, that is division of nucleus. So, every cell contains nucleus. Nucleus is the hereditary character. The DNA, RNA, everything present in the nucleus. So, division of nucleus is known as karyotype. Karyotype, in the 3rd paragraph, it's see, page number 266. Karyotype is the number, size and shape of the chromosome in the cell nucleus of an organism. Okay. What is karyotype? It says the number, size and shape of the chromosome. In a cell, the number, size and shape of the chromosomes can be determined by karyotype. Clear? Now, DNA, deoxyribonucleic acid, which is the important factor. Now, let me rub the board and see. DNA. In the human beings, we can see only DNA. deoxyribonucleic acid. So we used to say, suppose there is any doubt in the parentage, suppose sometimes in the pictures we have seen, even in the real life we have seen, male may deny that he is not the father of the particular child. In that case, we can go for DNA test. Okay, so DNA test, you would have heard about it. What is DNA test? That is the molecules of our parents from the mother and father, they are present in our body also. Oh, they just brought to our structure and everything. So when either male or female, they deny they are not the parentage of the child, they can go for DNA test. By DNA test, it is confirmed either she is the mother or father of the child, because the same DNA molecules only present in the younger cell, the younger organisms also. So parental chromosome, parental DNA and children DNA both will be same. So by finding the DNA test, we can analyze whether he is a father or not. So DNA, it is the hereditary material and it contains genetic information. It is hereditary material. DNA is hereditary material. Okay, I said already deoxyribonucleic acid. It is the most important part of the chromosome. This is DNA, double helical structure. What are the points about DNA? Double helical structure. Two layers joined together. It is first found by James and Crick. James Watson and Francis Crick. So short we are saying James Watson only, James Watson. This is model of James Watson and Crick. So they only gave the structure of the DNA. So they got the award for that. What are the chemical composition DNA? What are the things present in that? So this is double helical structure, you see. This is double helical, two lines are there. So here it is made up of nothing else, it's made up of sugar and proteins, phosphates and proteins. So what are the things present in the DNA? Guanine, cytosine, adenine, thymine, like that. Okay, so this is what are the things present in that, what are the components made up of DNA, we'll say. It contains sugar molecules. What are the things present in that? Sugar molecules. What sugar? Oxyribose sugar. Molecule. Sugar molecule. And then nitrogenous base. There are two types of nitrogenous base, that is purines. Nitrogenous is further divided into purines and pyrimidines. These are all very important, children. The purines you can see adenine and guanine. The pyrimidines, cytosine and thymine. So, I said sugar molecule, sugar phosphate molecules, nitrogenous base, guanine and cytosine. That is together known as what? Pyrine and pyrimidines. The pyrine you can see adenine and guanine, and pyrimidine, cytosine and thymine. G for guanine, C for cytosine, adenine and thymine. So, you would have got a clear structure of DNA. Let us continue in others class.